So we were approached by a early stage biotech startup with more than 70 unique, submitting more than 70 unique bispecific antibody drug candidates of various formats for rapid characterization. The main objective was for us to determine the bispecificity of each drug candidate. Bispecificity is an indicator of its purity and developability. Together with this, they also wanted to identify some of the abundant PTMs or post-translational modifications of the 70 unique bispecific antibodies, trying to screen if any of these are important, presumptive CQAs, critical quality attributes. And so the MS data package that will be generated will be used to back up the election of their top candidates for GLP tox animal studies. The turnaround time for this particular exercise was 2 weeks from submission of samples to preliminary results. There is a huge challenge when working with a large number of samples, and that is the main fact that one platform method for bispecificity will not be applicable to all samples. And the reason behind that is, of course, the fact that we are working with different bispecific antibody formats. And each of these formats has unique structural properties, and they will require different intact mass analysis MS-based workflows. So here on the small table on your left, we've summarized, what we have done. We had to make sure that all of our intact mass workflows are in good shape, that they can address the needs of each of these bispecific antibody formats described on the table. We have different versions of the knobs and holes. And then we also have the first generation bispecific antibody knobs and holes, or what we call the KIH. We've also encountered knobs and holes that are supercharged. And then the most complex version of the bispecific antibodies that we worked with are the multimeric ones, wherein we had to be more creative on how we can derive at most accurate bispecificity numbers. So here we want to highlight how we've utilized native LCMS and how we came to this decision. Here's an example of the raw mass spectra of a supercharged bispecific antibody, the panel at the top, compared to the panel at the bottom, which is a deglycosylated intact NIST antibody. As you can see, the raw mass spectra for the deglycosylated supercharged bispecific antibody is more compressed, and the M over Z range, the M over Z range is lower, much lower than your typical deglycosylated IgG1 sample. The supercharging limits sensitivity and selectivity for what we intended to do, which is to differentiate the major species from the two minor species of this bispecific antibody. So we thought about two different ways of navigating this challenge. One was to remove the acids in the mobile phases in order to minimize supercharging. We also thought about changing polarities, but it's a good thing that the first bullet point worked. What we did was remove the acid, work closer to neutral pH so that we minimize the charging effects and shift the M over Z range to higher M over Z range where the multiply charged species are more properly spaced apart. And so as a result of this, we were able to calculate bispecificity of our supercharged bispecific antibody samples. And here's an example of the deconvoluted mass spectra showing the different percentages of the major species and the two minor species. And so with native LCMS being able to solve this issue, we went on and started analyzing over 70 bispecific antibodies, determining their bispecificities. And here they are, 16 samples of knobs and whole format and the multimeric format, showing the main species of species 1 and the 2 minor species, and also for the multimeric forms of samples we received, looking as well for the bispecificity of species number 2. And then the rest of the samples of over 50 samples were analyzed using native LCMS. Again, most of them are supercharged molecules, and so they were very helpful. Native LCMS is very helpful in determining bispecificity, again, differentiating the main species from the minor species of these bispecific antibody samples.